and I thought the immune system was complicated enough. Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klanowski, your host for today's episode. Surveys show that public speaking ranks high on the list of fears of most people. Our guests today have embraced the challenge and come through with flying colors.
Our guests today are Hannah Ericson, doctoral student in the Department of Genetics, and Jordan Parker, a DVM PhD student in the Large Animal Medicine Department at the College of Veterinary Medicine. Hannah and Jordan respectively won first place and runner-up in last spring's 3-minute thesis competition at UGA. Hannah and Jordan, welcome to the podcast.
Thank you. Great to be here.
So today we're departing from our usual focus on infectious diseases to talk with Hannah and Jordan about the 3-minute thesis competition. So I guess the first thing that we should cover is, what is the 3-minute thesis competition? So Hannah, starting with you, what is the competition and why were you interested in entering it?
Yeah, so the 3-minute thesis competition is a really cool opportunity to get to talk about your research with people who you wouldn't normally get to talk about it with. So the goal of the competition is for you to distill your research down into a 3-minute talk that's aimed at people who know nothing about what you're talking about. And I heard about the opportunity through an email, and I was really excited because one of my favorite things we do every semester in my lab is we do a meeting where we talk about elevator pitches. So if you had 30 seconds in an elevator to talk about your research with someone, how would you do that? So I thought this seems like a little bit larger scale to talk about your work with someone completely new, and I thought it sounded really exciting and fun.
So Jordan, I knew you took a research communication class. So is that what prompted you to enter?
Yeah, so we actually talked about the 3-minute thesis specifically in my research communications class, and one of our assignments was to create a 3-minute thesis. We didn't have the timing worked out so that we could present the 3-minute thesis we made in class at the competition, but I think they've actually changed the class so that now students who practice their 3-minute thesis in the class will have the opportunity to present it in the competition. They've aligned the timing so that the students get that opportunity.
That's great. So I actually also teach a science communication class, and I also have the students do a 3-minute thesis. Some of them enjoy it and embrace it. Others are not happy about it. But, I actually tell them you need to have multiple pitches. You need to have a 3-minute. You need to have a 60-second. You need to have, and at pitched at different levels. So,
And you also have to have a pitch that really spans different audiences, right? So you should have one that maybe is important for the general audience, like you guys were doing for this competition, but maybe for scientists in general, maybe scientists in your field, right? So there's kind of different levels of audience as well. So what did you guys find the most challenging aspect of the 3-minute thesis competition? So we're going to ask both of you. So Jordan, we'll start with you this time.
Sure. I would say that studies shown that most people are afraid of public speaking are absolutely correct. That is certainly the case with me. I get super dry mouth and my palms get sweaty, but I wanted to challenge myself. I think speaking is a very important part of science, and being able to communicate your work, like you said, to a lot of different audiences can really make or break you as a scientist. So I really wanted to put myself out there and try my hand at communicating my work to somebody that maybe had no experience with the kind of things that I do.
Oh, well, kudos to you. So, Hannah.
Yeah, I think one of the hardest parts for me was trying to figure out all the information that I needed to cover before I could actually get to talk about my research. So I found either talking at a conference, talking to people in my department about my work, there's like several different points you have to hit before I can actually start talking about my work. So they're on the same page as me. And so trying to figure out what exactly I needed to talk about and how long I was able to spend talking about those was, I think, the hardest balance for me, because of course you only have 3 minutes. You want to leave time to actually talk about your research, but you still got to give that background information. And if I may contribute, I totally agree. Picking which information is important to share and what you should be nuanced and nitpicky about versus what you can kind of glaze over is so difficult.
So did you guys find that pretty challenging? And I guess to follow up with that question, what you thought you should have as background when you were first starting to prepare for it, did that end up being the background that you ended up using, or did you make some pretty drastic changes?
Parts of it, I kind of made mine into a game. I wrote out on little sticky notes like different pieces of information that I knew that I needed to cover, and I put them up on the wall in my living room. And I'm like, let's kind of make a map of what we're going to talk about. And so as I was going along and trying to construct the story that I was going to tell, I'm like, okay, we can take that one out. We need to add these 3 right here.
So Hannah, I have to ask, did you connect them with red yarn?
I thought about it. I could not find any yarn in my apartment when I was doing this. I have green ribbon. You can use green ribbon. No, I think that's a really clever way to go about that. I went about it a little bit differently. I created an analogy first. I wanted to be able to compare my work to something that people were familiar with. And so I based the information to include on what fit nicely into that analogy, but that centrally made sense to what I was studying.
Fascinating. I'm already learning how to
I know.
consider presenting my own scientific research.
So I think what we do with all of our guests is we ask them about their pathway to science. And so we'd like to do that with you guys too. So Hannah, how did you decide to get into science and go to graduate school?
Yeah, so I didn't really know what I wanted to do even in high school, like my junior year. And I just happened to take an AP biology class that was being taught by a fairly new teacher. But he was doing something really interesting where instead of lecturing at us the whole time, he gave us these packets. And so we would go work with our groups on the packets and figure out different biological concepts basically by analyzing a figure. And I got really excited because I really loved working with my friends and getting to kind of teach myself about what we were talking about in class that week. So I'm like, yeah, let's do biology. So I knew I was interested in biology, interested in science. I didn't really know what I wanted to do with that. I actually got my bachelor's degree at the University of Iowa. And while I was there, I actually signed up to be a tutor, a private tutor. They were a really cool program where if you get a high enough grade in a course, you can sign up to be a tutor. And I loved that. I had so much fun tutoring people on intro bio and intro chemistry. So I really learned that I loved to teach people about science in particular. And also I was a part of a group that took personalized DNA testing, like 23andMe and Ancestry DNA. And we learned about science through our results and also went and taught members of the community about their own results. So there again was another way that I learned I really love talking to people about science and teaching people about science. So I knew that I wanted to teach. I knew I also didn't want to teach at a high school level because my mom teaches high school. And I've seen the drama that is involved. And I'm like, I don't necessarily want to deal with high school students. So I ended up applying to the ILS program here at UGA, which basically lets you rotate through any of the departments that's involved in the program. And I started rotating actually in an entomology lab. And as I was looking for my second rotation, I was talking to a student in that lab. And he's like, well, if you're looking for somewhere else to rotate, my other lab we're looking for people. I'm like, your other lab? What do you mean? So he introduced me to the world of biology education research. And literally within an hour, I was on a Zoom call with my now PI talking about projects that they had in her lab and what I might be able to do. And I fell in love. I had no idea that you could do research on education and how to best teach biology to students. But that is absolutely what I wanted to do. So I kind of lucked backwards into my PhD research, and I've never looked back. I love it so much, and it's been so exciting for me.
That's very cool.
That's super cool. I think we actually have kind of a lot of overlap there. I also loved my AP Bio teacher. My mom is a bit of an inspiration for my journey, and I initially wanted to be an entomologist. Yeah, that's so crazy.
Oh, wow.
Yeah. So when I was little, I wanted to be an entomologist. My mom used to yell at me all the time for bringing bugs into her house. She was not having it. But she's a veterinarian. And when I decided that I wanted to pursue veterinary medicine, that was pretty early on and definitely inspired by her. I then went to undergrad at the University of Kentucky, where I majored in equine science. And I was fortunate enough to meet a really awesome lady named Dr. Emma Adam, and I did some undergraduate research under her.
during COVID. So it was mostly playing around with Excel sheets. Turns out I love formatting Excel sheets. But Dr. Adam was a fabulous mentor, and she really encouraged me. So when I applied to vet school, she also encouraged me to apply to be her graduate student. So when I got the offers from vet school and then for a paid master's position, I was like, free degree, they're going to pay me. I'll go back to vet school. It'll be fine. So I went and did my master's under Dr. Emma Adam, and she was amazing. She was such a cool mentor. She really valued communication and connecting our work with other people, but she also worked really hard to build me up and encouraged me to pursue a DVM PhD. So DVM is the Doctorate of Veterinary Medicine, and then of course the PhD is the science part of that. So I also kind of stumbled backwards into a science career, but I just loved my master's work. It was so fascinating and so nuanced. There were so many avenues that I could think about and explain the things that I was seeing or why I wasn't seeing results that we thought we might see. And I really liked that mental jigsaw, if you will, of trying to put together information that has been cultivated under these very specific conditions and has to be interpreted with great care because of that, right? So now I'm here and I am in the DVM PhD program in the vet school, and I'm really enjoying it.
You know, what I'm hearing from both of you is a lot of enthusiasm and passion for your research. And I think that ties into the 3-minute thesis. If it's presented as the passion of your life, something you love doing, you love talking about. So it's no wonder you guys were winners.
Thanks.
Yeah,
I mean,
absolutely.
If you're not excited about what you do, how are you going to convince anybody else to be excited about it? You know?
Exactly, right? The audience can feel you when you're speaking, that's for sure. So we would be remiss if we didn't give you both a chance to talk about your doctoral research. So Hannah, why don't you tell us a little bit about your work? And I promise that we will not cut you off after 3 minutes.
But you cannot use a laser pointer.
Okay.
Okay.
I did not bring a laser pointer.
Okay,
So I think
thank
we're
God.
Yeah. So there have been a lot of calls nationally to improve how science courses are being taught at the college level. Not to say people aren't doing a good job, but there's been a lot of really great research on how we can incorporate things like active learning or different techniques we can use to help improve student experience and student learning. But it can be really hard to convince faculty to start to employ those things in their classrooms, just because they can be hard to learn and maybe a little uncomfortable. But one of the ways that we can kind of help convince faculty to start to consider using some more of these like active learning techniques is through how teaching is being evaluated. If you evaluate teaching in a way that is able to recognize and reward the use of evidence-based teaching practices, then people, if they're being evaluated that way, they're going to be more motivated to start to incorporate those things into their teaching. Unfortunately, most teaching evaluation systems are not set up that way. Most universities use only student end-of-course evaluations to evaluate teaching, which are those surveys at the end of every semester that people are asked to fill out but don't always fill out. And these surveys don't really tell us anything about student learning. They actually don't give great feedback for teachers, and they're known to be really, really biased against certain groups of instructors, like female instructors, marginalized individuals, people where English isn't their first language. So these surveys really aren't the best way to be evaluating teaching. A better way to do that is to incorporate more sources of evidence. So using not just information from students, but also using things like a peer evaluation, where you have another faculty member come in and evaluate your teaching, or doing a self-reflection where the instructor themselves reflects on their teaching and what goals they may have and how they can improve over time. So my work is a part of a project that's here at UGA that's been working to improve teaching evaluation across the university. It's called the Delta Project. And one of the ways the project works is by working with department heads. So department heads are really key players in teaching evaluation because they're the ones who often are doing the teaching evaluation or at least organizing it. So we've been working with department heads to kind of think about how to improve teaching evaluation in their units. And so my work has been looking at, has teaching evaluation been changing over time as a result of our work with these department heads? And in the cases where it has, what are some of the things that are kind of affecting whether or not people are starting to implement these new practices or why they may choose not to do anything.
So one question I have is, you want to improve teaching evaluation, which will hopefully improve teaching effectiveness. How do you measure improvements in teaching effectiveness?
Measuring improvements in teaching effectiveness is a little more difficult, but there are some really great tools that are out there that you can use. Things like pre and post tests. There's a lot of really great tools that have been published that you can use to monitor student learning throughout the class. So you test them at the beginning of the course, you test them at the end, you can see how they've improved. Looking at student learning outcomes and what they are reporting that they learned in the class is another really great way to do that as well.
So do you think that part of graduate school education should include teaching in public speaking?
Oh, absolutely.
And do you think that this could help the future professors or the future people who are teaching these classes?
Absolutely. I think you talk to most faculty members and they'll tell you that they never were taught how to teach. Exactly. There really isn't a lot of that training. And so I feel like if you put a little more emphasis on it in graduate school, people can start to learn how to do that a little bit earlier in a more supportive environment. So there is a course when you TA here at UGA that you have to take to learn how to be a TA. And it's great. My instructor was great. I learned a lot, but I feel like there was still more learning to do. So I actually just completed the grad teaching certificate that's offered by the CTL here. Because I'm a grad student, I have more of these opportunities and I have a little bit more time to do that. And so I think anyone who's at grad school can really benefit from learning how to do that, especially if they are thinking about maybe going into academia later, because I think a large part of being a scientist is teaching the next generation of scientists and communicating your work. Science is inherently a collaborative thing. You're always building on other people's work. You're sharing your work, what you've done with other people. And so if you haven't really developed those skills, you're kind of at a disadvantage. So I think it would be really beneficial for people to get more practice with that while you're in grad school, when you have a little bit more support from other people and you have a little bit more time.
Yeah, I think, that's going to take a little bit of a change, right? Because we have this kind of negative connotation with teaching, like, oh, I'm going to have to teach, right? From the graduate students, especially ones that don't see any benefit maybe of taking that or learning those skill sets. And then you also have to have PIs that are willing to give their students the time and the space to be able to hone in those skills. So it's an interesting concept. So I want to ask another question that's, more of just a general interest for me, I guess, is, do you believe that student learning practices are set early in one's educational journey? And do you think that we should be thinking about some of these teaching outcomes and practices at an even earlier stage? So, for example, in high school, is anyone looking at the same thing essentially you're looking at, but maybe at an earlier time frame?
Oh, absolutely. There's so many people who are doing education in the K-12 world. I'm a little less familiar with that area just because that's not where my research has been focused, but absolutely the things that you're learning in high school, like one example I keep seeing show up on my social media feed is like how to study. Mm-hmm. You see people giving tips on TikTok all the time, like these are the best study techniques. And if you learn how to study one way in high school, that's probably how you're going to do it continuing on into college. And anecdotally, the way that I learned to study in high school is not the way I needed to study in college to be successful in my classes. And I just didn't really have the know-how and like how to effectively study when I was in high school. I didn't really need it. I wasn't being as challenged in those courses as I was in my college courses. And so when I got up to my upper level genetics courses in undergrad, I was like, oh no. I had to really quickly learn how to study effectively. And not everyone has the resources that I did. Not everyone can figure that out as quickly as they need to. So I definitely think starting to look at how students are learning, how they're studying earlier will really be beneficial to people coming in to like college.
Okay, so Jordan, let's hear some about your research. And I know you work on stem cells. So I guess the first thing I should ask you is, what is a stem cell?
Yeah, of course. That's a great place to start. So a stem cell is a specialized cell found in everyone. Everyone has stem cells. So from the very beginning of conception, you have totipotent stem cells, so cells that can turn into any tissue of the body, as well as any tissue of the placenta or the amniotic sac, etc. As your embryo grows, you start to get pluripotent stem cells. Pluripotent stem cells can turn into any cell that you would find in the body. From there, you get multipotent stem cells, and those stem cells can turn into any of the tissues in one of the 3 germ layers. So the endoderm, so your inside tissues, if you will, the ectoderm, the outside tissues, and the mesoderm, so the tissues in the middle.
So they start getting a bit more specialized.
Correct, as they go along. So my work focuses on multipotent stem cells because we can find multipotent stem cells in adults. And I specifically look at stem cells that reside in the bone marrow. So there are stem cells all throughout the body. There's some in the bone marrow. You can get them in the fat. You have neural stem cells, which was actually a really cool discovery, several years ago now, but we used to think that the brain wasn't able to regenerate itself, and this stem cell population suggests otherwise. But these stem cells have this kind of magical property, if you will, and that they can differentiate, and also they direct the activity of other cells. So stem cells will sometimes respond to injuries and coordinate the cellular activity of your immune cells, so your white blood cells, and that includes things like neutrophils. So it might tell neutrophils when they should send out their nets to capture bacteria or maybe when to back off from doing that. It might tell macrophages, which engulf bacteria and secrete a lot of inflammatory signals. It might tell it to maybe talk a little less loudly and secrete less of those inflammatory signals and kind of calm everything down. So that's really where the focus in stem cells lie, is in their immunomodulatory capabilities. So old school thought that you used to be able to give stem cells and they would engraft directly into the tissue. And that's not to say that they don't do that. We just don't have really strong evidence that they do that. But we certainly have strong evidence that they kind of are conductors of this immuno response to things.
And I thought the immune system was complicated enough.
Right? The immune system, oh man. I was like, how do you avoid that? You can't.
Thank you. As a card-carrying immunologist, I will say thank you.
No, it's very cool what stem cells can do. And while I think they're super fascinating cells in terms of what they do once they reach a site of injury or disease, I'm more interested in how they reach that site of injury or disease. Because the process of using stem cells for a treatment involves taking the stem cells out of their natural context. We then grow them in the lab and then administer them back to a patient. And that may be administering them as whole live cells. Mm-hmm. That may be taking off their cell media and purifying the little particles that they release into the media and administering those. That may be incorporating the cells into a scaffold so that we can put it in an implant or use it as a wound dressing. But when we go to administer whole live cells, we put them into the bloodstream or we put them directly into the injury. And it turns out the cells don't maybe love being put directly into the injury, but also some injuries you can't directly inject, right? We would never inject you directly in the hoof if you were a horse because we can't get a needle in there. So we instead administer those directly to the bloodstream. And they have to go through so many obstacles in order to then make it to the site of injury that we think that they should be going to. And so I really want to understand what those obstacles are and what obstacles the stem cells don't need help clearing and which ones they struggle with. And so that's what I'm really interested in, with, of course, the ultimate goal to benefit the horse.
Right. And I think we haven't made that quite clear yet, is you're looking at this in an equine system.
In an equine system. So as I mentioned earlier, my undergrad degree was in equine science. I am...
And you are from Kentucky.
I'm actually originally from Georgia.
Oh, okay.
Yeah.
Still in Kentucky. Okay, I thought there was a horse thing there.
No, I did my undergrad and my master's at Kentucky, though, so I definitely spent a long time there, and I would be lying if I said it wasn't because they're a big horse state. But no, I'm a card-carrying horse girl, and just applying that enthusiasm for the horse in a in a science way, if you will.
Right. So are you focused on trying to figure out what the mediators are that get these stem cells to a certain place so that maybe you can provide them, right?
Yes.
That's the intervention you're looking for.
Yes.
So where are you at on that research? Do you have any ideas or any candidates at this point?
Yeah, so when I presented my 3-minute thesis, I was actually presenting my prospectus. So I think that's really cool that Hannah and I have that difference. She's a little further on in her program as far as I understand. And I was like at the end of my first year when I gave that. So that was a cool opportunity. I'm specifically interested in 2 cytokines, transforming growth factor beta or TGF beta and interleukin 6 or IL-6. These cytokines are actually very highly expressed during hoof inflammation. So horses, any horse person will tell you they're beautiful and they're exceptional athletes. We did not breed them for their intelligence. So they're quite talented at hurting themselves, specifically their legs.
Right.
They got those beautiful long legs and they never know where they are. And so, like I said, TGF beta and IL-6 are hallmarks of hoof inflammation and actually kind of unique to both horses and people. So TGF beta is more expressed in the limbs of the horse than it is in the body of the horse. And that can lead to something called exuberant granulation tissue in horses. And in people, those are called keloids. I'm interested in how these specific cytokines that are a signature of a specific injury help attract and recruit these cells.
So, Jordan, I know you're early in your project, but have you observed positive effects in the horse from the stem cell treatment? And how do you measure it?
Yeah, that's a great question. So in veterinary medicine, we're certainly regulated like human medicine is, but our regulations are definitely a little bit different. So stem cell therapies are actually already clinically being used in the horse. And there are lots of in vivo studies, so studies that are done in the horse, looking at positive effects of stem cells on a variety of conditions, particularly wound healing. And we would measure that by looking at the rate of wound healing, for example. So if we make an injury on the skin in one leg and we put like a stem cell dressing on top of it versus an injury in the other leg that we don't put a stem cell dressing on top of, we can compare how quickly those wounds heal. Like I mentioned with the exuberant granulation tissue, you can look at how fast that fibrosis is forming. So the fibrosis is the organization of the tissue as it's healing and the extracellular matrix. So scar tissue looks a little bit different than normal tissue. So you can actually compare the scar tissue of a wound that's been treated with stem cells to the scar tissue of a wound that hasn't been treated with stem cells and see differences in how the collagen is ordered, which is really interesting, and test the elasticity of that and the strength of that tissue. It's definitely difficult to measure. It can be elusive for sure, but we don't see very many adverse reactions, for example. There's not a lot of hypersensitivity reactions, which makes sense. Stem cells don't express high levels of the receptor that would make them prone to causing a hypersensitivity reaction.
And in each case, you're pulling stem cells from the same horse it's going to be delivered back to?
Correct.
Okay.
So the way that one of the regulations that the FDA has, it's FDA guidance for producers number 218, I believe, specifies that we can only use autologous or cells that originate from the animal that we're giving them to. So at this point, there are no allogeneic, so cells that come from another but same species cell source for humans or animals. So autologous cells are the way to go.
So your research is animal focused, and you, surprise, surprise, have clearly articulated why you are interested in looking at this in a horse. But is there anything we can learn from your studies in horses to apply to stem cell therapies for humans?
Absolutely. Actually, a fun fact about the horse is they're the recommended orthopedic model, animal orthopedic model, for human orthopedic diseases. So they get injuries to their joints and their musculoskeletal systems in a lot of the same ways that we do, because we do use them in these highly athletic contexts, but also in the way that we administer the stem cells, the number of stem cells we need to administer, and how we grow up the stem cells is really similar between horses and people. So we would administer the stem cells to the horse in a limb vein, just the way that your doctor would administer stem cells to you in your arm vein. And we collect these stem cells from the crest of the hip, which is the same way your doctor would collect stem cells in a person. And we use them to treat those orthopedic injuries that I mentioned were pretty common between people and horses. So there's a lot of overlap there. And I definitely think that learning about maybe the way that these things behave differently between species can help us better understand how they work in one species or another.
Okay, as opposed to 3 minutes, in 3 words, what should scientists consider when they wish to effectively communicate?
Yeah, I think the 3 words I would use are context, audience, and through line. So I always like to think about talking about my research as though I'm telling a story. So you want to make sure that that through line is clear and that people can understand and follow you. And with that, with the context, you need to make sure that you're giving the background information that they need to follow that story along with you. And then audience, I think it's really important to think about who you're talking to and what they may need to understand to fully follow that story with you.
I think this question is going to illustrate exactly the beauties of the difference in thinking that Hannah and I have, but it's also the same. Like, I feel like our answer is going to be the same. So my answer is approachability, adaptability, and priority. So you need to think, again, about the audience that you're communicating to, and you need to be able to communicate in an approachable manner, something that's accessible to everybody that you're speaking to, in a way that is inviting and encourages people to take interest and to ask questions. And then you need to be able to prioritize which information you want to share. Like I said earlier, do you want to go deep down in the weeds and get really nuanced? Or is it okay if you kind of gloss over this and move on to the next concept?
I think those are both great answers. And I really like the focus on knowing the audience, because I think that's number one. You can't communicate effectively if you don't know who your audience is.
Mm-hmm. I think that's a big one. And I think the priority is a really important one too, because I think especially as you move through your graduate career and you've accumulated more knowledge and more data and more information, you want to show how smart you are, right? And you kind of want to vomit everything you know, right? And that's not effective, right? So, and you guys obviously were able to demonstrate that in the competition and here with us today. So our guests today were Hannah Ericson and Jordan Parker. Thanks for being with us.
Thank you for having us.
Yes, thank you for giving us this opportunity. Always fun to talk about your work.
So links to additional information about the 3MT competition at UGA can be found on our Instagram account at PPP Podcast UGA. We should note that this year's competition is November 14, so less than 2 months away.
Thank you for tuning in today. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, and special thanks to our production assistant, Sid Wigand, for research editing, scheduling, and keeping us on track.